Good morning and good afternoon to all of you for joining our live webinar today, a new technology to evaluate mature cardiac contractility on high throughput level. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be moderating your event today. Joining us as speakers will be Dr. Sonia Stolze-Fikes and Dr. Matthias Gossman. Sonia currently serves as the Director of Scientific Affairs at Nanion Technologies, and as of 2016, she is co-chair of the CEPA High Throughput Screening Ion Channel Working Group. Matthias serves as co-founder and CEO of innoVitro and is an expert in biotechnology and holds a doctorate degree in chemistry. Upon completion of the presentation, we will hold a live Q&A session, and as such, welcome you to ask our presenters questions at any time. You can log a question via the chat window found on the right hand of your screen. If you experience any technical difficulties at any time, please let us know via the chat box on your right hand side of the screen. Today's presentation will be made available on demand shortly after the event. I will now turn over the presentation to Sonia for the start of the presentation.
Okay, thank you very much, Jason. Hello, everybody. I'm happy to introduce to you today a bit of Nanion Technologies, and of course we clearly have a focus today on CardiacCyte and FLEXcyte. So I will start off with some general information on CardiacCyte and show you some recording examples and some capabilities, and then Matthias will turn over to some FLEXcyte data. So for the beginning, a brief introduction to Nanion Technologies portfolio. So since about 18 years, really our core technology is automated patch clamping. We started off with the Porter patch. Now have the Porter Patch Mini available since last year, which is really cool, I think, because the amplifier is integrated into the system. We do have higher throughput systems, patch liner and synchro patch for example, 384 recordings in parallel. Then we have some bilayer recording technology. This is the Orbit family. This is what you see on the lower left part here. So here you can measure single channels of reconstituted proteins in lipid bilayers. And for example, the Surfer technology, this is what you see on the lower right side, has the capability to do recordings of electrogenic transporters. And then, of course, we have the CardiacCyte and the FLEXcyte technology, and this is what we will focus on today. So the CardiacCyte system comes with a controlled environmental chamber. So the system basically is sitting outside of the incubator. It has 96 well consumables, which are long-term stable, and the electrode design sort of allows you to do parallel impedance and ESP recordings. The system also comes with an optical and electrical stimulation capability, and furthermore, the impedance mode allows you not to only look at contractility of cardiomyocytes, for example, but also to look at proliferating cells like cancer cells or hepatocytes. So the system comes with a liquid handling robot to help out with cell seeding, compound application, or medium exchange. And basically, taking all this together allows the system to be a perfect tool for example, cardiac safety screening. So next, I want to just briefly show you the hardware and software capabilities of the CardiacCyte and also FLEXcyte system here. So, like I said, FLEXcyte, the basic system allows you to do impedance and EFP measurements of the same cells in one well, and sometimes it is really of advantage to look at both signals of the same cells, because only if you look at both signals, you sometimes do capture the real and true compound effect. For example, also compounds decoupling contraction from the electrophysiology, like myosin 2 blockers, can be nicely investigated here. So what you see now on the screen is the graphical user interface of the software FLEXcyte control, and you basically have an overview of the 96 wells in, for example, mean beat. On the right side, you see 16 parts highlighted as a zoom in. Sorry, we're just having some technical difficulties here. Okay, I think we are now back with the slides. I hope you could see them still. I just continue with the presentation here. So again, graphical user interface, which allows you to capture with one glance your 96 well and the beating shape or the impedance traces of your experiment. So speaking about mean beats that we typically capture within the experiment. So a mean beat is sort of calculated by averaging a recording sweep. So we calculate the mean beats for impedance and EFP traces. And the advantages here are basically you have a higher signal to noise ratio, and the calculation of parameters is more consistent. You get the info from the entire well, so you don't have to pick sort of a golden electrode, and you do have a better overview of your experiment. This is an example. So you see a full recording sweep on the left-hand side in control recordings, and a mean beat calculated out of this. If you have cell arrhythmia happening, for example, here introduced by the application of the fatty light, you get sort of a arrhythmic mean beat, and you can easily capture by looking at the graphical user interface if something is happening with your cells or not. So coming to the parameters that are captured in the experiment, there are quite a lot which are captured while the experiment is going on. This is our so-called online analysis. So you get base impedance and beat rate or beat rate regularity. We also capture the primary and secondary beats, which sort of show you arrhythmic effects, and furthermore pulse width, amplitude, and so on and so forth. So similar parameters are also captured in the EFP traces, also beat rate regularity, amplitude, FPD0 and FPD max values, and so on and so forth. When analyzing the data, we have a really nice software part which is called DataControl. So here On the left-hand side, for example, you see the EFP spikes, sort of the sodium spikes in the presence of control and increasing concentrations of tetracaine. And on the right side, you, for example, see a sample IC50 recording of these tetracaine measurements. And in principle, with DataControl, you can pick any parameter that you like and calculate either IC50 or EC50, or also like percentage of block. So in general, for DataControl, this is a software that we make available for different platforms that we have. For example, for FLEXcyte and SynchroPatch are also the server platforms. So I think this is quite nice. So actually the license provided with our platform is not user limited. Furthermore, the analysis templates can be stored and shared to ensure consistency. And furthermore, DataControl can be installed on as many computers as you need. So this makes it really easy to share data and allow certain users to analyze or look at the data. Right, so let's have a look at some data examples now. Here I have some data from the myocyte validation study. Some on the left-hand side, a high-risk compound, disopyramide, which is like a negative inotropic compound. You see in the impedance trace that the amplitude is going down and the field potential duration is increased. So it's also a potassium channel blocker. And tamoxifen is not doing too much with the contractility, but is reducing the field potential duration. Domperidone is a medium risk compound, which at the highest concentration is leading to a cessation of beating. And another effect, for example, is that the sodium spike is reduced, so it's inhibiting the sodium channels. And these data were basically collected with different groups. So this was a cross-site study, and the acknowledgement can be seen here on the right-hand side. All right, I don't want to go into detail of this here. This is just an overview of the percentage of change of the FPD values. The only thing that I can say here is that the results were pretty similar to Ksenia Blinova's paper, which was also published during the same time, capturing the MEA data. Right, I don't want to miss also showing you some cell arrhythmias that were captured during the study. So we could capture basically all types, both in the EFP and also in the impedance mode. And Matthias will actually show you some cell arrhythmias as they have been recorded on the FLEXcyte. So another cardiac measurement, another example is this one here that I'd like to show you. This was a nice study where we looked together with some other groups at two compounds, sofosbuvir and amiodarone. So these are hepatitis C and antiarrhythmic compound. And if you apply these compounds alone to iPS cardiomyocytes, then there is no effect neither on the contractility nor on the electrophysiology of the cells. But if you apply them together to the cells basically stop beating. So on the lower left side you see a reduction of the beating amplitude, but they continue to fire. So the electrophysiology is still going on in the cells. So this is what you see on the lower right side. And this is just an example that shows you that in principle it's really an advantage to have both recording modes at the same time. So let's have a look at some pacing capabilities. We made a nice experiment where we took some cells on cardiac site sensor plates and paced them for 48 hours. And these cells nicely responded to isoproterenol. So this is what you see in the green bar. If you compare that with cells from the cardiac site plate without any pacing, you do not see much of an effect with isoproterenol. And if you have the cells on a FLEXcyte plate, you do actually also see a nice response here. Matthias will also go into details of this one a bit later. Right, and as another example here, so this is a pure impedance measurement, not with cardiac cells, but with cancer cells, breast cancer cells. This was actually performed in the University of Göttingen, and this was a recording which lasted over like 500 hours, so this is really many days. And what they did here is they treated the cancer cells with some medication, and you nicely see, for example, that the cell growth was reduced. And after washout, this is basically the second vertical line that you see in the graphic. The cells started to regrow, but only the ones who get a very low concentration of the compounds. And what I want to actually show you here is that the cardiac site sensor plates are really stable, also over weeks, so you can plan very long recordings with them. So with that one, I'm coming to the last two slides now. So like I showed you, with the cardiac site you can do EFP recordings and impedance measurements. And the FLEXcyte is basically an add-on for the cardiac site system. So it's in principle a different lid which the user can easily exchange. And with that one, I would like to transfer my presentation now to Matthias, and he will be able to show you some more on the FLEXcyte system.
Well, yes, thank you very much, Sonja, for the nice talk and for the opportunity to speak here today. I hope that everybody can hear me well. Actually, before starting with the FLEXcyte technology itself, I would like to start with some considerations on contractility of cardiomyocytes and their mechanosensitivity. So basically, the state of the art when it comes to contractility measurement is still the Langendorf heart and related ex vivo techniques. The Langdorf heart actually was first described in 1895, but is still the gold standard. And that although it has a list of drawbacks, of severe drawbacks, like the low predictivity, as you usually do not use human organs, but more likely animal models. It has a very low throughput and is very labor intensive, as you always can just use one organ per experiment. But still, this technique does have a big advantage, and that is its physiologic responses, basically. And we believe that this derives from the natural mechanical environment that the cells have. And if we look on the cellular or tissue level, different things can happen to a cardiomyocytes depending on the actual state of their environment. So if you imagine that you have your cell in a very soft substrate, just like a gel or so, what the cell will do, the cardiomyocyte, is it can contract one time and it will contract maximally, and that is what we call isotonic contraction. The other extreme is isometric contraction. Here, the cell have like infinite rigidity of their surrounding environment, and so that's exactly what happens if you seed the cells on very hard substrate like glass or plastic, and there we have this maximum stress that the cells go through with each cycle of contraction and relaxation. So they are going from one extreme to the other. But under physiological conditions, actually we have oxytonic contraction, which is basically the physiological relationship. So here the cells have a certain kind of mechanical resistance in their surrounding, in their environment, and that actually withstands the contraction and makes a positive feedback loop between force, stress, tension, and the mechanical resistance. In fact, the cells above that are able to sense actively what's happening in their surrounding. So basically, cardiomyocytes do sense their surrounding by use of deformable adapter proteins. The protein that they obviously use the most is talin, and they use it to sense it by fast muscle myosin contraction on the one hand side, and slow non-muscle myosin contraction. And the deformation of different adapter proteins actually regulates the differential protein kinase pathways. So if the cells are if the surrounding is too stiff, the talon will be stretched too much, and if it's too soft, the talon will be not stretched enough, like you see on the lower right side. Above that, they are mechanosensitive on a gene expression level. So actually, cardiomyocytes respond to their unphysiologic matrix stiffness with drastic transcriptional deregulation. So here we see the results of a group from University of Cologne, where they seeded the cells on soft substrates like 12 kilopascal, medium substrates with 30 kilopascal, and very stiff substrates with 123 kilopascal, and in addition on polystyrene. And what we see on the lower left side is that when the cells are seeded on very stiff gels or on polystyrene, a big portion of the genes are deregulated. So we have 604 and 800, while when the cells were seeded on very soft gels, it was just like 7 genes that were deregulated. And what I personally found most striking in this study actually was that it was basically cellular metabolic processes that were affected. So it's not just the cytoskeleton or other processes which one might think are very relevant, but it's also cellular metabolic processes that actually determine the fate of a cell very deeply. Another factor in mechanosensitivity is, of course, the ion channels of cardiomyocytes. So here we have some results of a study from 2 cells and had a look on their calcium spikes when the cells were mechanically stimulated by uniaxial stretching. And what's quite obvious here is that with increasing magnitude of stretching, the calcium influx actually increased as well. And obviously, like you can see in the middle plane and on the right side with silencing RNA for L-type calcium channels, it's exactly those calcium channels which are stretch sensitive. And this is really crucial because this is one of the most important channels in cardiomyocytes. And if they are stretch sensitive and you have your cells on a very rigid substrate, a big portion of those channels will not work properly or not work physiologically. So the basic challenges that we have if we come to the market and want to replace animal-based ex vivo models is, of course, the predictivity and the physiological relevance. But additionally, what we see regularly is, of course, we have to increase the throughput quite a lot and need to be very cost effective. And that's how we came to the point with this technology. We call it the FLEXcyte technology. So this is an add-on to the CardioXcyte. But in this case, the cells are cultured on ultra-thin silicone substrates with adjustable mechanical environments. And those substrates with the cells on top of it are deflected downwards by the weight of the medium on top of it. And so every time when the cells contract simultaneously, the membrane is lifted upwards a bit. And when they relax, it goes down again. And actually, the cellular stress can be calculated from the deflection. And that's also quite a simple formula that's used for that. So here on this slide, you can see how it really looks like. So on the left side, you see the top view of the 96-well plate seeded with iPS-derived cardiomyocytes. You can see the membranes go up and down with your naked eye. And on the right side, you see a bottom view. And there you can also see the membranes moving up and down. So as this technology is an add-on to an impedance-based system, people always ask us, what is the basic difference between impedance or CardioXcyte and the FLEXcyte? So the main parameters that are different is the substrate, of course. So on impedance-based systems, you have an infinitely stiff substrate. So the contraction that the cells can exert there are basically isometric contractions, which are not physiological. And the parameter that is recorded here is basically the dynamic changes in cellular shapes. So here with impedance, as you know, this is the resistance of a high frequency field. And so the basic parameter is the change in the resistance based on the change in cellular shape, so dynamic changes. While with the FLEXcyte, we use basically quite a lot of the electronic part, but here we have very physiologic stiffness, and hence we have oxytonic contraction, and that's also the basic parameter that we measure. So really the contraction, the contractile properties of the cells. But as I said, the electronics is very similar, just that we use impedance to measure contraction, real contraction. Here on the right-hand side, you see one mean beat that we record with this system, and the parameters that we capture. So of course, the blue line on top. The amplitude is a very important parameter for contractility, but it's not the only parameter that is of importance, basically. So we also have the durations of upstroke in green and downstroke in red. We have the corresponding slopes of those phases, of course, the integrals, and what we do not see here, but what is also measured, is frequency and arrhythmic events. So to validate this technology, we measured quite a lot of compounds. I would like to show the basic benchmark compounds that we found out seem to be very important. So for acute compound application, this is obviously positive inotropes, because positive inotropes are not so easy to measure with IPS-derived chitomyocytes and standard assays. So we chose here for this presentation isoproterenol, a beta-adrenergic agonist, SBA, which is a calcium channel opener, and omecamtiv mecarbil, a myosin activator. And in all three cases, if you can see on the traces above, they all exert positive inotropic effects. In the lower panel, you can see the actual beat amplitude. So in all three cases, the beat amplitude rises with increasing dose. This is all in short-term, so acute experiments, like up to 30 minutes. But what you can also see, if you take a look on the durations, basically, that although these are three positive inotropes, their actual action on the shape of the contraction is different for each of those. So with isoproterenol, with increasing concentrations, both the duration of upstroke and downstroke decrease. So the actual mean beat will shorten. In contrast with SBA, the upstroke duration increases with increasing concentration up to a certain concentration, then it goes back again, while the downstroke duration actually increases quite a lot and then decreases a little bit. So here we have very contradictory effect, while with omecamtiv mecarbil, the durations are not really altered in nanomolar doses, but if we come to the micromolar regime, it's especially the downstroke duration which is affected a lot, and this actually this effect even increases with higher concentrations. But then comes a concentration range where the cells really cease beating. So here you can see the effects actually differ a lot depending on the actual substance rather than the substance class. To benchmark our system, of course, we also want to do chronic compound application, because not all the compounds are working within minutes. So for a lot of compounds, you need more like days and weeks until the onset of the adverse effects. That's why we chose kinase inhibitors and anthracyclines, exerting structural and/or functional cardiotoxicity. And here we decided to have compound incubations of 5 days. So usually we have 5 to 7 days of pre-culture, where the cells attach properly and start beating synchronously. And then after that, we did 5 days of incubation with compound and monitored the cells every day, starting 2 hours after compound addition. So here we see 3 different compounds. On the left side, allotinib, which is supposed to be a non-risky, non-cardiac risky compound. And that actually is reflected by our measurements. So in the only in the micromolar doses you do see an effect like a 10 micromolar, which is very super physiologic and super clinical. With 1 micromolar, there is an effect after 3, 4, 5 days. But in general, in the nanomolar range, we do not see any significant effect with this compound. While with vendetanib in the middle, the effects were seen already after 2 hours. So at the highest dose of 10 micromolar, the cells even ceased beating for 2 days. And on the second day, they started beating again with a very, very low amplitude. So it was really hard to measure that. What we can see here with this compound, that the results are very stable over the whole time. So this compound actually has a very quick effect, but a very stable effect. And that is in contrast with idarubicin on the right-hand side, where we see very dose and time-dependent effects. At the highest dose, again, the cells cease beating and they do not start beating again. With the lower concentrations, 1 micromolar and 100 nanomolar, we see a gradual decrease within 3 to 4 days. And with the lowest concentration of 10 nanomolar, also a decrease. And after 5 days, the cells did not even stop beating. So here we can see both time and dose-dependent effects. Also with those compounds, we had a deeper and closer look on the beat patterns, on the beat shapes of the cells, and especially on arrhythmic events. And here we could see already after 24 hours different kinds of cell arrhythmias, how you can call it. So first of all, beat rate irregularity. So in this case, and that was true for all the wells that we had with those with this compound, actually the cells switched between fast beating and very slow beating back and forth for several days. I've never seen this effect before, so and I have no conclusion on what that might be, but it was really a consistent and very interesting effect. What we can see also is after contractions, of course. So here marked with red arrows. So in this case, these were really extra contractions with low amplitude, but you can see that it was really irregular because the distance between the beat before and after that was different to those usual regular beatings. And in the most extreme case, we could also see fibrillations where the cells did not arrest really. So there was beating, what we can see here, with quite a high amplitude, but that was really irregular and possibly decentralized. So we should have a closer look on that. But these are actually the 3 patterns that we could see here of different kinds of severity, basically. So to sum it up, to conclude, we believe that the FLEXcyte system really provides additional physiological relevance, as the cells really, cardiomyocytes here can sense the mechanical environment that they would sense in the human body, and their structures and their parts can work as they should. We have quite a high throughput with 96 wells, and hence have quite a good cost effectiveness. And additionally, which is really nice, it directly integrates into Nanion's accessories, especially the temperature control and CO2 incubation systems. So you do not have to operate your system in the incubator, but you can operate it as a benchtop device. So it also integrates into the Soul head for optical pacing, which we also did already, and which works really nice with channel rhodopsin 2. Really handy, and yeah, so it really integrates very well to the existing system. So to conclude it up, actually, if you are interested in using such a device in your lab, feel free to come to Nanion and speak about it. Actually, we were planning quite many demos in the upcoming days and weeks. So basically, these plans need to be postponed a little bit. But in the meantime, if you have drugs that you have to need to have screened very soon, just come to us, and we deliver the service for you. So I would like to thank our partners from Nanion, like Ulrich, Sonja, and Elena, who are really working very closely with us and very intensively, and of course from University of Applied Science Aachen, Professor Staat and Alexander Jung and Aravind. And of course, I would like to thank the cell providers, which were very generous with the cells, and we could do a lot of experiments. So that's it from my side. I will now hand over back to Jason, I would say. So thank you very much.
Thank you, Matthias, for your presentation, and Sonja for going through the introduction to today's event. And thank you to all those in attendance that have brought the questions forward. I will now transition to Sonja to address some of the questions that we've received so far. And again, we will address all the questions, and we will transcribe the event and make it available on demand.
Okay, thank you. So we have quite a bunch of questions coming in here. So I will just start with the first one here. Are FLEXcyte plates transparent? Can one use them to multiplex assays such as calcium imaging? So the answer would be yeah, in principle, yes. The flexible membrane is transparent, and the thing is, due to its geometry, it can only be focused on different areas of the membrane with an optical system. But, well, it worked already in high contact imaging devices, for example, with calcium-sensitive dyes. That was quite robust regarding focus changes, but I would say this needs to be checked with the respective system that one wants to use with the transparent plates here. Another question is, can I work with any other contracting cell type, smooth muscles or skeletal muscles? So in principle, anything that contracts with sufficient force could be recorded on the FLEXcyte plates. And we are currently working on testing different muscle type cells apart from the heart. So I would say everybody stay tuned for any upcoming application notes here. Right. Another question would be, have you observed any changes in expression patterns of cardiomyocytes when grown on FLEXcyte plates in comparison to rigid substrates? And here our answer would be like, well, at the moment we are talking mainly about phenotypical responses that we capture. But we hope to get more information on the expression levels, on the protein levels that are expressed in the membranes. So this is also an ongoing project. Maybe another question here. How do you compare FLEXcyte to 3D models? So in principle, by co-seeding cardiac fibroblasts, we can achieve three-dimensional structures. And the 2D culture has the advantage that comparably low cell numbers are required. And And yeah, basically, when using fibroblasts, you could come to a 3D model type, cell type, basically.
So thank you, Sonia, for addressing some of the questions that we've seen come through the Q&A. We would like to thank you all once again for joining the event, and we will transcribe the questions along with the on-demand webinar, and that will be made available through our main website as well as all our social media channels. And we will conclude the event with this